For joining us today in this seminar by Dr. Petra Vaiglova. I see everyone in this room, and I assume many people online joined us also yesterday for Petra's all-day training in statistics. Today, we will see a very interesting case study where isotopic analyses have been used to examine boars from the Zagros region. But what is very interesting about today's paper and what Petra will analyze is how, by focusing on the dental microstructure, we can increase a lot the resolution of the information we can get about ancient herding practices, diet, etc., etc. So I will not reintroduce Petra, right? I mean, she's brilliant. She came from Australia. She's a pioneer in isotopic research and statistical analysis. And we're very, very happy that she's here with us. This seminar, like yesterday's workshop, is part of the One Archaeological Science project funded by the European Union in the context of the ERA chair funding scheme. And we will have many more coming up in the following months, so stay tuned. Petra, on to you. Thank you very much again for being here.
Well, thank you so much for that lovely introduction. And thank you for having me. I'm absolutely thrilled to be here to both talk about training in statistics and wild boars. This is the first time I'm talking publicly about this case study. It came out about a month ago, so I'm really excited to finally share kind of everything that we've done over the past couple of years working on this. So let me move my next slide. Okay, so I'm going to kind of bracket the talk about the actual case study with a little bit of insight into what we know about teeth, how we use teeth in isotopic analyses, and how maybe we can increase the resolution of what we can measure. So this is where we are, kind of just taking a broad step back. We're looking at how isotopic values from tissues, we can measure human tissues, we can measure animal tissues, how they contribute to our understanding of past dietary practices, mobility practices, perhaps some paleoclimatic patterns. So when we measure teeth, and I'll be specifically focusing on teeth today, so everything is about teeth. When we do isotopic analyses in teeth, we usually look at two different isotopic proxies. We look at carbon isotope values, so that's the delta 13C, and stable oxygen isotope values, delta 18O. Carbon usually gives us an indication of what the dietary input was that individual was eating during the time of tooth formation. And oxygen gives us an idea about the sources of water. And this could be both water that the animal or human drank directly, or that came from the plant or the food that the individual was consuming. So we're getting these two different proxies. We're usually measuring it from the enamel. Inside the tooth, we also have the dentine part. We can measure the dentine, but it doesn't preserve as well. So usually enamel is kind of the gold standard of what we're measuring. Now, the most common method, and I've done this a lot myself, the most common method of measuring enamel is to kind of use this sequential drilling pattern. So this is a method based on the work by Marie Ballas, and everything that I say today, I have to preface with saying, I don't dislike this method. I use this method myself. I'm just going to kind of give some examples of ways that we can increase the resolution of it, but this is not a bad method. I have to make that very clear. So with this drilling pattern, I call this the macroscopic kind of hand drilling method. We make some assumptions about how teeth grow. They generally grow in this direction. So the enamel closer to the chewing surface, which is over here, is older than the enamel closer to the root. And so we go in with these hand drills and we take a sample about every 1 millimeter. So we've got a sequence of these 1 millimeter spaced increments of powdered enamel that we extract. And then we measure the stable carbon and stable oxygen isotope values of each incision. And then we build this beautiful curve that represents changes in the isotopic input of carbon, oxygen during the growth of the tooth. So we're able to look at seasonal patterns, at changes to the intake in the water, intake of the food. And these kinds of intra-tooth sequences, so when we have more than one measurement per tooth, have been really, really beneficial to understanding seasonality of climatic patterns, but also mobility patterns of animals. So one of the things we can look at is the grazing. Oh,
how long time-wise?
It depends. It depends on what tooth you're actually analyzing. So if you're doing a first molar, it's about a year. If it's the second molar, it's about maybe 18 months. But what I'll be showing a couple of slides where we actually go in and count the number of days that are represented in the enamel. So that's kind of the highest resolution that we can do. But generally, like when we do this, I call it the low resolution method. We're taking a really big step back and kind of taking the whole growth of the tooth and then making interpretations about like the first year or first 2 years of the animal's lives. That it's a very early part of the formation process. So the types of questions that we can answer have to do with grazing patterns. So this is an example. It comes from my PhD where I looked at some sheep and cattle from the site of Makrialos, and I measured some sheep from both like feasting context and habitation context. So animals that were used kind of for these feasting purposes, but also ones that were consumed on a more daily, day-to-day basis. And I was interested to see if their management strategies may have differed because they were used for different purposes. And the type of analysis I was doing is I was looking at the shapes of these intratooth sequences. And the reason I was interested in the shape is because we know how the values of plants, how the values of rainfall vary throughout the year. So this is just an example of some tree cellulose that has been measured. I think it's in the US. It doesn't really matter what the location is, but what I'm trying to show is that the way that oxygen isotope values and carbon isotope values fluctuate is driven by some climatic factors. So in temperate regions, it's usually driven by temperature. So you can see temperature go up and down, and so then the oxygen and the carbon isotope values follow. That's in temperate regions. In other regions like monsoonal or tropical regions, the variability in the isotopic values is not so much driven by temperature, but the rainfall amount. So we really have to pay attention to where we are in the world. But we know how the values generally fluctuate throughout the season. And then we look at what's happening inside the tooth of the animals that we study. And we look at these shapes, we look at the amplitudes, kind of the difference between the maximum and the minimum values of each sinusoidal curve, and infer aspects of seasonal management. So these specific sheep from Makrialos, what I inferred is that they were basically staying put at the same altitude. Their values vary predictably, just like the plants and the water from the region around Makrialos does. And they probably didn't cross altitudes. They probably didn't go very far from the site. And that's not what the cows were doing. I'm not showing the cows here, but they had slightly different patterns. Their sinusoidal curves were a little bit noisier. So that to me indicated that maybe they were grazing in a wider catchment. They were eating plants that had more variability kind of inside them. So overall summary of the slide so far is we're looking at the shapes of these sinusoidal curves to look at grazing patterns. Now, when the curves change, often we find these flattened curves. It could be for a series of reasons. One of the reasons we get these flattened curves is animals moving between the highlands in the summer and the lowlands in the winter, which is a very common transhuman pastoral strategy. That's something that we've observed ethnographically, and we know it's been happening at a lot of different sites. The reason the flattening is actually happening is because the animals are they're sampling, let's just talk about oxygen right now. They're sampling rainfall from the highlands in the summer, and that rainfall has lower values than rainfall in the lowlands, right? So they're sampling this. Oh, the cursor is different here than over there, so I have to look over there. So they're sampling this water at high altitudes in the summer, and that water at low altitudes in the winter. So we don't get as pronounced as sinusoidal curve, but it's a little bit flattened. So we've seen these sequences a lot. Transhumance is one explanation. The animals could also be eating fodder that was collected in a different season to the one in which the animals consumed it. But I'm starting to get a little bit hesitant about how we explain these flat sequences, because I suspect it might have a little bit more to do with drilling than we are actually taking into account. So if we take a step back and consider this drilling strategy again, what are the two main assumptions we make? We make two assumptions. The first one is that teeth grow linearly. They start here, they end over there, and it's just a linear, very, very nicely predictable pattern. The second assumption is that teeth grow at a constant rate. So an equal amount of enamel is just being deposited throughout the formation of the tooth. And it turns out that neither of these two assumptions actually hold. And how do we know that? We know that if we look inside the teeth and study their microstructure. So here is the tooth of a wild boar that we studied. A lot of this histological work that I'm talking about has been done with my colleagues in Germany at the University of Hildesheim. So that's Horst Kierdorf and Karsten Witzel. So we have a wild boar third molar over here, and we sectioned it at where we see that orange line. That's the sectioning plane that we decided where we're going to cut the tooth. Once the tooth was sectioned, now we're looking at the cross section of the tooth. So the enamel is up at the top. It's more white, and the dentine is on the bottom. It's this ground section is 50 microns thick, and it has to be that thin because otherwise we wouldn't see the growth structures as easily. If it's any thicker, the daily growth lines, they just overlap too much. So it has to be very, very thin. Now, if we zoom into specific parts of this cross section, so if we zoom into here and there, and we really have to zoom in, the scale bar on here, it's 100 microns, and the scale bar on the image at the top is 200 microns. So we're looking at this under the microscope. We can see the shapes of the daily growth lines. They grow like trees. So trees have their annual growth rings. Teeth have daily growth laminations, and we can look at the shape. We can measure their thickness. We can count how many days are represented in the growth of the enamel crown. And importantly, we can use it to kind of guide more targeted isotopic analysis. This is a really nice image that was also obtained by my histological colleagues. And that's a modern boar, a modern wild boar that was injected with calcine injections, non-toxic, at specific days of its life. So it was injected on day 86 and day 114, day 135. And those specific daily growth lines light up because of the calcine in the body of this animal. So we can see more clearly exactly the shapes of each daily growth line. And what worries me there is that the lines aren't horizontal. We would expect them to be horizontal if the growth was completely linear. Each daily growth line would be like this, but it's not. It has this kind of arched pattern and also slows down towards the end. So a small incision here represents a lot more days of growth than a small incision at the top. So that means that if we go in with these big incisions, each drill hole actually represents a different temporal unit, right? The one at the top might represent 60 days of growth, and the one at the bottom might represent 100 days of growth. And for some studies, if we want to not be as high resolution, and the high resolution analyses are very expensive, it still works. It gives us an indication of the changes in the isotopic inputs, but we have to pay attention to the fact that each data point is actually a different amount, like it homogenizes a different amount of enamel. And that's something that's really important to pay attention to. I'm gonna take a sip before I slow down a little bit. So just to summarize the approaches that we can take. So this is what I'm calling the macro hand drilling method. It's this low resolution method. It's got some pros and cons. So the pros are that it's cheap and fast and less destructive. All that we have to remove is some powder from the surface of the enamel, and we can measure it, it could be $10 per sample per one measurement. The cons are that the sequences have no temporal meaning, right? We're not actually following the patterns of growth. We're homogenizing a big amount of enamel growth, and each point is a different unit. And there can also be intertechnician variability, right? Different people might drill to different depths, or they might drill around the tooth, which is homogenizing a lot more days of growth than if the drill holes are shorter. So that's also something we're not really paying attention to. I think it would be great in the future to just post images, make a repository of the images of all teeth that are analyzed, because we have no way of really comparing how people's drilling strategies compare. So that's the macro handling method. We can do in situ analyses, so that's what I'll be explaining in my case study. This is done using the shrimp. This is the secondary high resolution ion microprobe, and it can target very specific parts of the enamel. So if we create a nice thin section, we can place these target analytical spots, which are about 20 microns wide, and we can place them on specific growth lines. Usually, it's best to do it close to the enamel dentine junction, where the enamel is mineralized, kind of it's best quality mineral. So we can do it targeted in a targeted way. The pros are that this technique takes the growth geometry into account, and so the sequences have more temporal meaning, but the cons are that it's very, very, very expensive, and it is more destructive because we have to cast the whole tooth in epoxy, which is really strong glue, and the tooth will stay in that glue forever. So if that's not something we can do, then the high resolution method might not be available. Some people try to use crystal bond, which is a wax. So they put the crystal bond wax on top of the tooth before casting it in epoxy. So technically, because crystal bond can dissolve, they are able to take the tooth out from the epoxy. That method hasn't worked for me yet. I need to experiment more. And yeah, the polishing also is really tricky because 50 micron polish, it's not that easy to do. All right, so low res, high res. Now I'd like to show you this case study that we published recently, which is on provenancing wild boars from the site of Asiab in the Zagros Mountains in Western Iran. So this site has been immensely important in zooarchaeology. It's been excavated since the 70s. It's one of the sites where that really allowed us to understand the morphological changes as we move from wild boars to domestic pigs. So it is the early Neolithic, but we are at a pre-agricultural stage. The boars themselves are still wild. But the interesting thing about the site, which is located over here, there it is, Western Iran. The interesting thing about the site is we have this really big feasting deposit. So there was a circular structure at the site, and in the middle of the circular structure was a pit that was called the boar pit, in which 19 boar skulls were interred, and they were placed very neatly, the mandibles to the one side, the cranial caps to the other side. So very neat deposition, very fast deposition. And now we have 19 boar heads there, which have butchery marks, so they were used, at least some of them, for consumption purposes. They may also have been used for sacrificial purposes. If you want to read more about the boars themselves and the measurements that have been done to understand that they're wild and not domestic, this is the paper to look at. So that the kind of core zooarchaeological analysis has been done by Pernille Bansgaard in Copenhagen. So what did we already know about the boars before the isotopic analysis started? We knew that they were wild, morphologically. We had both males and females represented in this pit. We had varying ages represented in the boars as well. Interestingly, wild boars were not the most commonly hunted animal in this region at the time. So there's some wild boars from early Neolithic assemblages in the Zagros, but it's not the most common wild animal. So having 19 of them in one pit, like that's special. We know that the crania had cut marks, processing evidence. The male skulls had the canines extracted, and it's interpreted that they were used as necklaces or for to kind of show status, that sort of a thing. The weight of the edible product has been estimated. It's hard to do. We have so many ages. We have both males and females represented, so there's big plus or minuses on these estimates. But about 350 to 700 kilograms of meat, edible product, from all these boars. So that could have been eaten by 350 to 1,200 adults, taking into account that people eat more on like a feast day than on a regular day. So that's a huge amount. That's not something that is consumed by people that are living at the site at the time. The villages there were way too small to fit, even 350 people. We're looking at much smaller kind of clusters of families. So even before the isotopic analysis, we knew it was a special deposit. We had butchery indicators, possible sacrifice. There's a lot of the skulls there, lots of different ages, both sexes represented. But what we didn't know is where the boars were actually coming from. We didn't know where they were sourced. All we had was that they were consumed, that they ended up in this pit. So that's where the main questions for this study came from. We wanted to know, where did they originate from? And because the proxies that we're using, because of equifinality, they can't ever like pinpoint one location where an individual is coming from. We were approaching it from the perspective of eliminating possibilities, right? So we knew we couldn't tell exactly where each animal was coming from, but we could eliminate the possibility that they all came from the same location, right? Maybe that they were part of the same like the family of boars that live together. We could also eliminate the possibility that they all originated from close by to the site. So the approach was kind of understand what's going on geochemically in the region, and then try to understand more about where the animals were coming from, seasonality of birth, and something about their dietary patterns that would allow us to see if they were coming from very dispersed parts of the landscape. So these are the teeth that we analyzed. We had two lower molars, so two second molars. They were both lower up here, and three third molars, one lower and two upper. These are the third molars. For the zooarchaeologists here, you're going to be excited about the aging. So we use two different methods for aging the teeth. One method is the one that zooarchaeologists use based on the wear patterns. So that's a paper that's been used for many years now to do the aging based on the wear patterns. We also did the aging, well, the histologists, my biology colleagues, did the aging based on the internal patterns, the growth lines, and what we know about what the growth lines show at different times of the lives of the animals. So that this column shows the age from the histologist, and that shows the age from the zooarchaeologist. They're similar. They're not identical, but they're very similar. And it's just because the two methods are slightly different. But it's good news that they don't differ massively. So in order to create these 50 micron thick thin sections, the first step was we wanted to, one, preserve the teeth. We were going to cut them. So we wanted to preserve them for kind of future analyses. So we did a micro CT scanning to understand kind of what they look like on the inside. Are there any micro cracks that we might want to avoid with sectioning? And where is the best place to kind of place the cut? Because not every cut will expose a nice section where the daily growth lines can be counted and kind of where they're exposed very nicely. So we scanned each tooth. This is the left tooth kind of from the top, from the side. And then we asked the software to kind of show us what, like the different slices of what the tooth looks like on the inside. And the enamel is the white part at the top, and the dentine is on the bottom. So enamel is denser. It appears white on the image. Dentine is not as dense, not as mineralized, so it's a little bit lighter. So now we knew what they looked like on the inside. So the next step was to cast them in epoxy. And then create these 50 micron thick thin sections, which were done by my colleagues, Horst Kierdorf and Karsten Witzel at the University of Hildesheim. This is an image that the press office at my university took. They were like, we're going to send someone to take a picture of you and the samples. And I was like, okay, but I've got thin sections. And they were like, that's okay, you can hold a thin section. So there's a nice image of me. I'm going to move it. Oh, okay, I can't move it. Interesting. No, sorry. Okay. So at the end of the process, we had these beautiful thin sections, which we could do histological analysis on, and which we could also put inside the shrimp and do some like trace element mapping, and also do in situ strontium isotope ratio analyses. So at the end of the histological analysis, this is what we had. Each tooth was mapped depending on what the enamel was showing. So these colors are, they're just guiding our understanding of how the tooth grew. So the very, very thinnest part in the middle, it's really hard to see. That's the dark green. That's the oldest enamel. And looking at this table, which was constructed by the histologist, we knew that the oldest enamel part, the green one, took 5 days to form. The next part, the light green, took 26 days to form. The next one, the yellow, took 27 days to grow. Orange, 63 days. So you can see the progression of the growth of this tooth, right? It's starting at the enamel dentin junction in the middle, and it's progressing in this kind of arched way. And you can, if you pay attention to this is the enamel extension rate. So that's how much enamel is deposited every day. At the start of the growth, 267 microns of enamel are deposited every day, but at the end, 38 microns. So this really shows us that So many more days of growth are represented here than at the top. It really slows down through life. So we really should be kind of adjusting our spacing. If we're doing sequential sampling, we should be adjusting our spacing accordingly. Otherwise, we don't have a sequence that kind of has equal increments. And so that's a second molar. It has 5 kind of sections that have been identified and where the length of the enamel density junction at each section was counted. And then we have the period, how many days and how much enamel is secreted. This is what analysis of the third molar looks like. It's longer. It represents 330 days of growth. So the first one, I didn't mention this, the first one, the second molar, this one, represents 197 days of growth. This one represents 330 days of growth. So it takes longer. We have a lot more sections that were, again, quantified, measured. We have the length of the enamel dentin junction in each section. We have the number of days that are represented and how fast the enamel is growing. So this is a summary of all 5 teeth. We kind of now have an indication of how many days are represented, how fast the growth is, and that's what we needed to do targeted high-resolution isotopic analyses. So this analysis consisted of 2 different measurements. We measured the stable oxygen isotope values on the shrimp, the sensitive high-resolution ion microprobe, and then we did strontium isotope ratios. Now, the shrimp spots, you can't see them because they're so tiny. They're 20 microns wide. If you really zoom in on this microscope image, you can see them, but otherwise, especially on the projection, you won't be able to see them. But because I knew how many days of growth were represented, I basically placed each target location about 7 days of growth apart. 7 is arbitrary. Our weeks are 7 days, but I could have done 10 days, whatever. I chose weekly resolution. So the spacing kind of started farther apart at the start and kind of was more compressed at the end, following the number of growth lines represented. So overall, I had between 23 and 44 samples per tooth. The lower end is for the second molars, the upper end is for the third molars, and they were spaced at weekly intervals of enamel secretion. Now, I should clarify that what I described about tooth formation was the first stage of tooth formation. So the secretion, the formation of these daily growth lines is just the first stage. There's another stage which starts happening after the secretion finishes, and that's enamel maturation, where kind of the enamel finishes its formation, it kind of gets all its crystallinity in place, and that wave happens later. And what that means for us is that the initially secreted enamel gets overprinted with a later dietary input, but it's kind of a later wave that happens kind of in one go. So that attenuates all of our measurements a little bit, but this is still the highest resolution we can get because we can't control for that later maturation kind of overprint. So that's oxygen. The measurements of the strontium isotope ratios were done on the laser ablation inductively coupled plasma mass spectrometer with the help of my colleague, Renaud Joannes-Boyot. And here we had about 19 to 23 measurements per tooth. They were also done kind of along the EDJ. They were spaced equally. And they're a little bit wider. So that's what the rectangles are representing. It's spots that are about 450 times 110 microns wide. So they're covering a lot more growth, but that's the kind of best we could do for what we knew about the teeth formation. And the last stage of analysis that we did is we did trace element mapping of the surface of the teeth. And this was to understand whether there are any dietary kind of differences between the animals, whether there are any events that we can identify. We measured lots of different elements, but the one we chose to focus on in the analysis was barium. So barium concentration varies in different food sources. We might be able to see nursing signals with it. A lot of people have been using it to kind of study the timing of nursing patterns. With the boars, we're just using it to see, do we have spikes in barium? Could they be seasonal? And how do these spikes compare between the different animals that we're studying? Now, the important part about reading these trace element maps is that they have to follow, if there's any patterns, which here there are, they have to follow the biogenic patterns, right? So we know from the histological analysis the shape and the geometry of tooth formation. So if our spikes are following that geometry, that's okay. If we see a blob, like if we had this massive red blob over here, that would be diagenetic. That would be the tooth taking up some barium from the soil. But if the spikes, if it's red, it's higher. If it's blue, it's lower concentration. The spikes, which we can see over there in the enamel, they are following the growth line. So that's not diagenetic because diagenesis wouldn't actually kind of do that uptake so faithfully. And plus, when it's sitting in soil, it's not cut in a cross section. So we did the last stage just to get an idea of what the diets of the animal were doing. So let's look at the results. So here are the results of the two proxies for mobility. So we've got the oxygen isotope values and the strontium isotope ratios. Those of you who are familiar with mobility studies, equifinality is a massive issue. As I said, we can't really pinpoint exactly where individuals are coming from. So the best thing to do is to use more than one proxy. That's why we use both oxygen and strontium. The good thing for us is that both of these proxies were showing the same pattern. So we had 4 boars that had overlapping oxygen isotope values. So the gray are all overlapping. And then this one individual, ASB 449, shown in the red, has much lower delta 18O values. It overlaps a little bit, but it's in a different region. So it's showing that this one individual is different from the 4 others geochemically. The strontium isotope ratios are showing the same thing. So this one individual in red has much lower strontium isotope ratios than the 4 others. And the measurement error is shown with the gray bars. The measurement error on the oxygen is also shown, but you probably can't see it because it's as wide as the actual symbols are. So the precision is quite high. So, okay, we've got this indication that 4 boars have similar geochemical signatures. One of them does not. Now, I spent a lot of time thinking about, those who were here yesterday, about statistics and how do we present quantitative data. One of the questions I really spent time thinking about is how do we actually describe differences, right? Some people would want to use a t-test or do some significance testing to like properly show that there's a difference between the red individual and the gray. I don't think that tells us much. Significance test doesn't tell us if the difference is big or meaningful. So a better way is to actually show the difference between the two groups. So that's what I did. For the delta 18O values, we reported what is the mean of the gray individual. So that's 133 through 407. We had 167 measurements. And the mean of the red individual, we had 88 measurements, was 2.8 with a confidence interval between 2.5 and 3.1. So the confidence interval is telling us that most likely the true difference between those two groups was somewhere between 2.5 and 3.1 per ml. And that's a big difference. That's interpretable, right? So we don't need a t-test to tell us that that's interpretable. With the strontium ratios, I didn't use confidence intervals, but the keen amongst you will notice that 2 sigma, very close, to a confidence interval. So with strontium ratios, the common method of interpreting non-locality is to set a baseline of values. The local baseline is usually what's measured plus 2 standard deviations. That's the local baseline. And then anything outside of it is non-local. So if we take that one group and say that they are the local range or local, maybe not necessarily to Asiab, but somewhere else, the red individual, it does fall outside of that 2 sigma kind of interval around it. So we're both proxies are showing us that something is different about that one individual. Now, the simplest explanation, which would not be contextually based at all, would be to say the 4 are local and 1 is non-local, right? That's the most kind of economically efficient way to interpret this. But we need to actually look at what's happening in the geology to understand what the values are doing. So this is a geological map of Iran. We've got the site. So Asiab is located over here. It's located on the Kermanshah plain. And another site where we do have some modern plant measurements. So that's the site of Ali Kush, which is located over here, about 100 kilometers like south, a little southwest of the site. Those are the only plant measurements we have for that region. So we took this map. We got some strontium isotope ratio values of rocks from this region from the GeoRock database and constructed an isoscape that shows the variability in strontium isotope ratios. Now, it's not the best map because it's limited by what we had. It's only showing what the bedrock is doing. It only has a very small amount of plants. Those plants from Ali Kush are over here. over there. But otherwise, we would really need to do a lot more sampling to have a more kind of well-defined map, and to also take into account more superficial sources of strontium, not just the bedrock. And this is the prediction error on that map. Now, important to pay attention to is what the geology is doing. At the site of Asiab, we've got Quaternary and Mesozoic, so that's younger geology. So over here, that's Quaternary Mesozoic. Ali-Kosh is Miocene, so that's older. So if we go back to our plot here, I said the simplest explanation would be to say that the four were local and one was non-local. That's the opposite of what the geology is suggesting, right? It's saying that Asiab is younger, so that should be younger. That one individual that's flagging up different looks more local as far as what we know about the strontium variability in the landscape is saying. So this one looks more local, and the four other ones are looking more non-local. So that's exciting. We have some indication that most likely they're coming from somewhere like not from the close proximity to the site. The program that my colleague Georgie Falster made to create the map of bioavailable strontium, or just strontium, not bioavailable, it is able to give us an estimate of what is the closest location where these values would be coming from. So the program says the closest location is about 70 kilometers away, which is a lot. Like that's a 2 days walk if you're carrying a boar. But we have to be really careful about that figure. We don't have a plus or minus on it because it's difficult. So the media, when they picked up on this, they love the figure 70 kilometers, but we really need to be hesitant about it because all we have with this map is the bedrock values, and there's more to strontium variability than bedrock. So we have to be careful, but it's not close. All right. Something else we were able to look at, because we had these high-resolution oxygen isotope sequences, is the timing of the animals' births. So this is a table that shows the timing of crown formation in wild boars, modern wild boars. We know, and a lot of this work was done by my histological colleagues, we know that boars are born in one of two seasons in temperate climates, so either in the spring or in the late summer. And what we have here is kind of an indication of when crown formation starts and when it ends. So the E is like early formation, L is late formation. I'd like you to focus on the second and third molars, because that's what we have with the boars from Asiab. So second molars are the orange, third molars are the blue. So for animals that were born in the spring, birth is in April. Their second molars start forming in June and in like January, and the third molars start forming in October and end in October of the next year. For the animals that were born in the late summer, it's pushed by a couple of months. So we know that the seasonal values, the delta 18O values of water, of rainwater, groundwater, plant water, it fluctuates seasonally because of temperature and evapotranspiration. So I put a very crude kind of indication of how it fluctuates. So we get high values around like July, August, low values around like January and February. Again, this is temperate climates in the Northern Hemisphere. I have to specify Northern Hemisphere now because I live in Australia. And so summer is like not now. So now that we know how the values fluctuate throughout the year, we could kind of compare to where it should be if the birth was in the spring versus if the birth was in the late summer. So these are the actual measurements. These are the intra-tooth sequences we measured for the second molars up at the top and the third molars at the bottom. So with the second molars, if the animal was born in the spring, we would expect the start of the sequence to be kind of close to the maximum, right? Starts around June, it should be close to the maximum. The end should be close to the minimum. It's, not approaching the minimum, but somewhere low. If they were born later in the late summer, their values would be kind of on the down curve, approaching the minimum, and then the end would be approaching the maximum. So that's what we did, kind of did this comparison. And overall, what it looks like is that all four, except for ASB 449, have the patterns that we would expect for a spring birth. So second molars, they're at the top, they're not close to the minimum. These ones, the third molars, they're kind of on an uptick. But 449 is doing something different. It's probably starting in the summer. So this is not the summer, and this is Also probably not the summer because summer is here and then approaching there. So there's a little bit of squinting involved with like properly reading patterns that are not completely smooth. But it looks like that Asiab ASB 449 was the one that was born in a different season to the other animals. And interestingly, that's that red individual. It's the same individual that has different geochemical values to the other four. So we're getting so many different lines that are indicating that it's spending its time of tooth formation in a different location. It was born in a different time of the year. And then we can look at what the barium, the trace element maps are showing. The analysis of the trace element maps was also done by the histologist who compared what we know about how teeth grow and where the daily growth lines are in modern wild boars. And they looked at the spikes. The spikes are a little hard to see on the projection, but they are there. So we have, wherever there's an arrow, we have noticed a spike in barium. So there's a spike around here. And so that's been estimated to be day 135 plus or minus a couple of days. Day 50, is it 55? No, 86 over here. And then there's a spike here, 330. It's not the darkest of spikes, but it is higher. The spikes are more visible in the dentine. So notice how these are very pronounced. They are biogenic because they follow the growth patterns in the dentine, but there also might be some later uptake just because of how much brighter they are. So we've got these spikes in the individuals, and they're happening at different times. So for ASB174, the spike was around September, because now we know when they were born, so we can estimate if it's day 165, that spike happened in September. For individual 360, spike happened around June, August. February, April. So there's variability here. And that indicates that the animals they're not kind of living in one group, like one kind of wild boar family moving around. They're probably dispersed around the landscape and eating different seasonal foods that are locally available. So it's just another angle to indicate that we're not looking at boars that were hunted close by to the site in one go, and that were kind of obtained using the most efficient hunting strategies in terms of like energy expenditure. So what do we have overall? I've got a beautiful image. So this was created by Catherine Killicky. She's a science illustrator, and she spent many months trying to digest all of the information that we found here. She created this beautiful image that has so much detail. It's amazing. She got the shape of the mountains right. Like when she sent it to me, she was like, so I put my dot on Google Earth and we're facing northwest from the site. So that's what the mountains look like. She calculated the distance to the river. She talked to the zooarchaeologist about what birds may have been there. Remember the canines? I said that the male canines had the male skulls had the canines removed. So we have an individual that has a canine necklace. We also have an antler that was found in the same deposit. So much detail, which is incredible. But what it's showing is this, the effort that the people are putting into transporting wild boars to this site. So we have an indication that they were not hunted close by to the site where they do live. The proximal environment is very hospitable to wild boars. They were living there, but the animals that ended up in this boar deposit that were used for the feast or sacrificial purposes didn't all originate next to the site. So they came from much farther away. There's different ways that this could have gone down. Either the people who lived here or lived close traveled to some other place to do the hunting and then brought it back. That's probably less likely than people from farther away who were coming over anyway, brought the gifts as like a gift, as a contribution to the feast. They could have also been consumed elsewhere. So the butchery marks didn't have to happen at Asiab. They could have been hunted and butchered elsewhere, and then the heads were brought over as trophies, like hunting trophies we know are used quite often ethnographically. But whichever of those scenarios it was, we think it's most likely that people were bringing contributions to the feast. Regardless of which scenario was true or closest to being true, we're talking about massive amount of effort that was involved in bringing the animals or even their heads back to the site where communal gatherings were taking place. That was really important for the region. I love this board, which indicates that like movement. Okay, we've got movement going on. Just for fun, I wanted to show you some of the headlines that came out. So I wrote a paper or an article for the conversation, and someone reposted it on Life Science with a new headline, which says, archaeologists discovered that parties 11,000 years ago were BYOB, bring your own board. Isn't that amazing? And someone made a meme on Twitter, on Blue Sky, with like from Simpsons. I haven't seen the Simpsons episode, so it doesn't mean much to me, but they're using BYO boar in this meme, which is great. And sometimes, well, oftentimes we talk about like the impact of archaeology, like what we do, how much meaning does it have for like the general population? I've got the answer. So these are screenshots of when this was in the news, and the article on the wild boars was sandwiched right between an article on global trade and healthcare insurance. So it's right there in the middle. And there's some other places where it occurred when someone was announcing these like what are they called? Like the devil horn volcanoes or something like that. So we were right under there. But that was really fun to see. So should we leave some time for questions? Yeah, okay, wonderful. So I'm gonna wrap it up there. I'm gonna pull up the acknowledgements. Thank you so much to everyone who contributed to this talk. Thank you for bringing me here. Now I forgot to add the logo. We'll add the logo, I'm sorry. I also have a paper on statistics I love to talk about. So here's my image on statistical training and the elephants that we don't talk about in the room. Thank you very much for listening. And yeah.
Thank you so much, Petra. This has been an extremely interesting paper and we learned so much. Questions from the audience first, or if there are any online. Can you check if there are any online,
Yes.
by the way?
Okay.
But for the moment, let's see what's happening in the room. Are there any questions in?
Yes.
Yes, so that the people online can hear.
Again, Petra, I really enjoyed the presentation. It's everything. All of this is close to my research heart. My PhD was on wild pigs, and I do isotopes as well. I was very interested in the shape of the growth lines, and I was wondering if you have any information about their shape or how easily it is to discern them in other species, especially caprines, if they are equally arched, etc. And then a second question is the interpretation. Usually in zoarchaeology with hunted animals, there is the tendency for heads to be missing from archaeological sites, especially if, with the claim that they came from as far as 70 kilometers. Logistically, we have observed many times that people choose to leave in the hunting spot the heavy, meatless parts, such as heads, which doesn't invalidate the interpretation, but it makes it even more interesting regarding the incentive that people had to bring back to the site the skulls.
Yeah, absolutely. Thank you so much. Great questions. I'm going to start sharing again, just so I have something to point to. I do have some more slides that if I had time for, I would have gone over. We're building this like simulation to understand teeth growth better. And there it is. That's the one I want to show. So for the simulation that we're doing, continue to share and close. I'm not seeing it there. Oh, interesting. Okay, it's gone. So I've only shown you the shape of the growth patterns in the wild boars. The shape in like sheep or goats, they're not the same. They're not as arched. They're not like this. They are flatter, like they're more horizontal, but not completely horizontal. So each of these red lines is that lamination that's been it's an isochron. It's 1 day of growth. So we've got them forming like that. So they're actually, they're not horizontal. They're not as overlapping as the wild boars. So if you went in with a drill and you kind of did an incision here, did an incision there, you're cutting across so many days of growth, which would mean that you might even be measuring very similar things across all the different incisions. That's not the same with a sheep. But there's still like homogenizing going on. The growth still slows down. So we have more days of growth represented in a later incision than the earlier incision. But yeah, there's been lots of histological work done on like what are the shapes, what's the timing, how many growth lines are there in like a second molar or third molar. I'm trying to find a way to do the shrimp analysis without having to do the histology every time because it's really time consuming and the 50 micron thick sections are really hard to make. So I'm trying to kind of find tables, like can we have estimates, like overall estimates for how many days are represented in a second molar from a sheep, and then use that to kind of come up with like an average spacing. Like maybe we put like 10 samples in the first third of the tooth, and then 20 samples in the next third, and 30 samples in the last third. So something like that. So that's the first question. Does that answer the first question? Okay, awesome. Thank you. And the second question was about the heads. Absolutely. It's so fascinating that it's the heads that are there. Because the butchering marks are on the heads, like there was butchering involved, but we don't know if it happened at Asiab or somewhere else. But like ethnographic literature shows that people bring heads as trophies, right? Like boar heads are a sign of maybe wealth or sign of prestige, or like if people are coming to this feast from far away, to make a contribution, they want to contribute something special. So like bringing that over could be like a symbol of kind of community building and social bonds. So yeah.
So the rest of
Not in this deposit, no. No. Mm-mm. Yeah, really interesting. Thank you. They might have been in a different part that wasn't excavated, right? Like this is just that one, the boar pit, which was inside the communal structure, was where they were really neatly located. So maybe the heads were deposited in a special way. They're kind of really neatly and deposited very fast. The soil was kind of put in really fast after the faunal remains were put in place. Maybe the other bones were somewhere else that we just didn't find. Yeah, sorry.
Other questions?
Oh, okay. 2 questions. Did you look at suitability of different teeth for enamel sampling? I'm thinking of either first or second mandibular incisors on mandibular male canines. I mean, we've considered it in terms of what we know about the growth of teeth. From this side specifically, we only had the molars available. It would have been nice to do canines because there's so much growth represented, so many daily growth lines there. So that would have been cool, but they weren't available from this site. And on the slide about the related ages to ground formation, is there any published information available on this? Yes, there is. There's a lot of work done by histologists. The main names to look for are Kirst and Uwe Kierdorf. Carsten Witzel is very prominent in this field as well. They are co-authors on this paper. So if you look up the publication for the Asiab study that I just talked about, they are co-authors there. But yes, there's lots of histological publications out there.
This technique is suitable for dentin too.
Dentate, yes, it is, but we don't know about the preservation as much. So someone's actually doing some experimental measurements now on using the shrimp to do nitrogen measurements from the dentine, because the shrimp can measure any element, right? We could only measure oxygen from the enamel because there isn't enough carbon in the enamel for the phosphate analysis, because like that 20 micron white spot, it doesn't have a lot of carbon, but you can measure anything. So there's been some experimental work done on the nitrogen. It's just we don't know if it preserves, right? Like is it original nitrogen or is it diagenetic? So you can do it, and we do know the growth patterns in dentine as well from the same studies that did the analysis of like morphology.
So sorry, it's just follow-up questions.
Yeah.
So it doesn't matter, like you're not affected by any collagen extraction from the dentine.
So this analysis was done just on the enamel.
Okay, makes sense.
Yeah.
Your talk overall, very well done, very enjoyable, even for a non-specialist. But I'm also fascinated by these daily growth rings or rates that you have. And I remember you said the second molar took 197 days and the third one 330. Now you had probably 8 second molars. Would they all have been given 197, or how big is the error or the variability there?
So they're not identical. Not every boar will have 197, but the error isn't that big. The error might be like 20 days. Even if it's 50 days, it's not, well, actually 50 is getting to a lot, like 50 out of 197 is a big proportion. But the plus or minuses they're there, but the individuals that have been studied, they were within similar ballparks. Yeah.
Or from a totally different climatic environment or a later in the year birth, would that affect how long it takes for the second molar to finish growing?
No. So the actual timing of birth, it doesn't affect the growth geometry. It just affects the chemistry. So depending on like what foods are available, but the thickness of the growth lines. The thing that really varies between individuals is the growth lines. They're actually, I made it sound easy, the quantification of them, but it's really hard because they're much thinner towards the enamel dentine junction. They're much wider on the outside, and the width also changes like from the top to the bottom. So to characterize them, it's like we have to trace these laminations. So like an isochron, like in one day of formation. That's why those individual sections, the color-coded sections, were drawn. And then the number of days was counted between each lamination along the EDJ. So that sort of thing might differ between individuals, right? The thickness might vary a little bit. So if you then measure like the width of the whole enamel segment, if you don't know how the thickness varies in individuals, you can't just divide by like, you expect there to be 50 days. You can't divide that and get your enamel extension rate.
And the last question on that, obviously, if you have a daily growth ring, there must be a gap in between when it doesn't grow. Is that in the night or is that in the day?
So
when does that deposition happen?
So the darker, like I like your thinking. It's very like tree ring oriented, like the darker and the lighter the part of it. There is no actual gap here, but the way that the mineral looks, it looks a little different. I think because of either the amount of calcium or oxygen. In the night, I think it looks a little bit darker in the night. And so fun thing that I didn't say about the formation of teeth, we can see the birth line, like in first molars, which I didn't have because first molars started forming in vitro. On the day that the individual is born, the natal line appears there, and it's much darker, I think, because it's so stressful for the body. Like the body does not want to leave. It's happy. So the birth is so stressful for the individual, so you can really see the line prominently because of all the stress. And I think there's like oxygen and things involved in there, like in terms of what it's actually getting. Yeah. Thank you. Wonderful.
First go, I can have a second one. Just to add a little bit, I guess there is a limitation in what age the animals that we sample should be, because with the wearing down of a tooth, obviously there is truncation of these growth lines and the signal. So very old animals would have truncated that duration of 190 whatever days. But regarding the interpretation of this individual that deviates from the majority, you have convincingly shown that the birth season might be different. Could the interpretation or the results be solely explained by that rather than different geology or distances from the site? For example, a wild pig that is born off season from the majority would obviously be constructing their teeth based on the differences in the seasonal diet of wild pigs throughout the year. Could that be used to explain it completely rather than involving distances from the site?
Great questions again. I love them. So first question was about the tooth wear. Tooth wear definitely is an issue. If we're losing like the top part of the tooth, we're losing a lot of daily growth lines. The great thing about the shrimp is because it's so targeted, the samples are like 20 micron wide, and we only need to place them close to the enamel dentine junction. Technically, all we need to get good measurements is the region close to the enamel dentine junction, right? So if you look at this tooth here, this is the enamel dentine junction in the middle, right? So if all of this is lost, if all of that's worn down, but you're still getting the region next to the EDJ, you can still place your samples there, right? The oldest enamel is the one over here. And that's actually when we do the hand drilling, we often capture the oldest enamel, which I didn't realize. So even if all of the top part is lost, as long as we have the EDJ and like an area maybe 100 microns from the EDJ, we can still do the analysis. So that's good. But then there's issues of like how well preserved it is, like maybe there's cracks or. Second question was about the interpretation of the red individual. The thing that makes me think that it's not just dietary or result of different timing of birth is because it's the whole sequence is different. So if we look at, I'm gonna bring back, oh, come on, bring back the image with all of the results. There you go. So all of the values for that individual lower. You know what I mean? Like if it was only a matter of the birth timing, it would be shifted, right? So like the start would be different than the start of someone else who's born in a different region, but the whole sequence is different. That makes me think it is geochemical, like it is based on the location rather than just timing, because if it was timing, it would just be shifted.
There is a question online.
Oh, yes. Okay. Zoom. Oh no, where am I? I'm lost. Uh-oh. Where's the Zoom?
Can you use the other laptop? Sorry.
Yeah, okay. Were you able to sample an end-to and free from the same animal and then overlap the results in order to provide a longer time range covering more than 1 year? Sorry. So the question was, did we have both a second molar and a third molar from the same individual? Not from the site. So we have it. One of my master students finished a thesis on looking at doing this kind of analysis in a horse too. So we have horses from Japan, and we had the first molar, second molar, third molar, and we're trying to get a long sequence where hopefully the overlap will kind of work out. It's not as easy to do. If the sequences were completely smooth, it would be a little bit easier, but in theory, it is possible. We didn't have that for this site, but that's something we want to do for the future. Yay.
No, anything online? Now is the time. No, I think
That's it.
is it? Yeah.
Yes.
Fantastic.
Thank you so much.
Okay, so thank you again very, very much, Petra.